class we have talked about the anatomy of a dicot root. Coming on to dicot stem, one clear and most important difference that comes up is in the case of vascular strand. There the xylem was exarch and here the xylem is endarch. Endarch means protoxylem is towards the center, metaxylem is outside. Clear? So let's begin with the outermost layer, which is known as the epidermis. In the case of dicot stem, epidermis is the outermost layer. In the case of dicot root, we saw epiblema was the outermost layer. So epidermis is the outermost layer. The main function is protective cover that is providing, and the cells are cuticularized. So cuticularization of the cell is what is seen. Next to it is the cortex. Now cortex is a thin wall of parenchyma cells, and similar to the dicot root, it does have intercellular space. So cortex does have intercellular space. Next to it, we have a layer of endodermis. Endodermis is the layer which is the innermost boundary of the cortex, and it is made up of barrel shapes, but this endodermis does not has casparian stripes as in the case of dicot roots. In the dicot roots we saw casparian stripes. Endodermis in the case of dicot stem does not have casparian stripes. But there is presence of medullary ray. Medullary ray are radial strips of parenchyma which are present adjacent to the vascular bundle, and these actually are bigger than the cortical cells. So again, an important layer. Hypodermis is the layer which is just below the epidermis, and this layer has collenchyma cells. So collenchymatous cells, we can say, are present in hypodermis, and this is due to the deposition of extracellulose material. The next is pericycle. Pericycle is a layer which is heterogeneous in nature. There is alternate bands of parenchyma and sclerenchyma. So parenchyma, sclerenchyma alternately arranged in the case of pericycle of a dicot stem. So again interesting. Next is the vascular bundle. Vascular bundle comprises of xylem and phloem. It is endarch because protoxylem is towards the center and metaxylem is towards the outside. It is ring shaped, conjoint, that is it has both xylem and phloem together. It is collateral, that means both are present on the same radius, and it is open, that means there is presence of cambium between the xylem and phloem. So that means it is open. Presence of cambium indicates that it is open. Now this is an indicative of the secondary growth that takes place in the dicot root. In the dicot root, we say fascicular or interfascicular cambium is present. Fascicular or interfascicular cambium, that means that you have phloem and xylem on the other side of it. Now these cambial cells are responsible for active division, and along with the cambium there are certain medullary ray cells which also become an integral part of the interfascicular cambium. Now these together, the inter and the intrafascicular cambium together form the ring of the cambium, which gives rise to secondary phloem on the outer side and secondary xylem on the inner side. So phloem on the outer side and xylem on the inner side. So development of secondary phloem and secondary xylem takes place, and then from the cambium, what arises is a layer of hypodermis later on. And then we do have a pith. The pith is integral part because it extensively develops the central portion of the ground tissue, and it has parenchyma cells, and this pith has presence of intercellular space. So again important to note that pith has a presence of intercellular space. So this was just a quick understanding and a summary of the dicot stems. Understanding each of the components here become extremely important because this is how we differentiate it from the anatomy of a dicot root. So just going through the structures, we can identify how dicot root and a dicot stem would differ. Specifically the vascular bundle here is ring, conjoint, collateral, open and endarch in nature. Specific characteristics you must keep in mind. So we'll be coming up with many interesting biology sessions. Stay tuned.